Next, we would like to have presentations of venture companies which have state-of-the-art technology. Presentations will be made by following speakers. Megakaryon Representative Director President Mr. KENICHI AKAMATSU. TWOCELLS Representative Director President Mr. MASAYA Matsumoto. Heartseed Founding Scientist Representative Director President Mr. KEIICHI FUKUDA. Orizuru Therapeutics Representative Director President and CEO Dr. Ph.D. Mr. NONAKA KENJI, and Shinobi Therapeutics CEO Mr. KEMP. These are the 5 speakers. First, we would like to ask them to give presentation, and the questions will be given by moderator Mr. Ando. Everyone, would you please come up to the stage? The theme is venture companies presentation which have state-of-the-art technology. Would you please come up to the stage? Now, Mr. Ando, the floor is yours. So this is a session of venture in regenerative medicine. Thank you very much for joining us. First of all, we have companies which have a very interesting technology. So we would like to ask each company to give us your talk covering your company, and then I would like to ask you some questions. Now, so should I follow the list that I have? First, President AKAMATSU of Megakaryon. I am AKAMATSU from Megakaryon Corporation. Thank you very much for giving me this opportunity today. Megakaryon is a company that creates platelet for transfusion from iPS cells and aims to commercialize transfused blood cell products that do not depend on blood donations. In other words, a paradigm shift in transfusion therapy. We started full-scale research and development after obtaining funding in 2013. Since then, we have spent about 10 years developing the technology to produce large quantities of iPS cell-derived platelet for clinical use, and in 2022, we succeeded in administering allogenic iPS cell-derived platelet to a patient for the first time in the world. In terms of management, we became a member of the Sysmex group at the end of 2023. As you know, transfusion of blood depends on donated blood, which cannot be imported. In other words, the stable supply of blood is a key factor in national security in the medical field. In Japan and other developed countries, the population is getting older, and there are concerns about the supply of blood. In Japan, 3.6 younger people capable of donating blood provided for one elderly person in 1990, but by 2070, one person will need to provide for one elderly person, a significant increase in the burden. In addition to such medium to long-term shortages, we have often experienced temporary shortages due to natural disasters or infectious disease pandemics. In particular, the supply of platelet is at the highest risk due to the short shelf life of 4 to 5 days and the inability to stock up on these products. Megakaryon aims to contribute to stable supply through iPS-derived platelet as much as possible. Let me explain our core technology. The main feature of our technology is that instead of iPS cells being differentiated into platelet, megakaryocyte cell lines, MKCL, which are the progenitor cells of platelet, are established and cryopreserved in a master cell bank. The production of platelets begins with the culture of MKCL from the cell bank. After the required amount of MKCL has been grown, the growth is stopped by changing the medium to induce a maturation and release of the cells. This method is very efficient for producing large amount of platelets and is the basis for our own practical scale production process that is ready for human use, which was uniquely established by us. The cell structure of the iPS platelet made in this way was observed with an electron microscope, and it has the same microstructure as native platelets on the right. After establishing the method, by conducting the preclinical studies one after another, after going through PMDA review, for the first time, we conducted the world's first clinical trial of allogenic iPS platelets. This is the result of administering 3 units, a third of the standard unit of Japan of iPS platelet to a patient with reduced plasma counts due to the aplastic anemia. And you can see in the red, after iPS cells were administered, the iPS cells increased in the blood and circulated for a certain period, which was reflected in an increase in total plasma counts of the patient body. Unfortunately, the clinical trial had to be terminated after this one case, but the fact that the circulating ability of iPS plasma was confirmed suggests that they can work in human body, and encouraging for a future development. In the future, Megakaryon will develop HLA and no HLA knockout platelets for use in patients. We call it universal ones with platelet transfusion refractory patients. At the same time, we hope to solve the problems currently faced by donated blood platelets by ensuring a stable supply, eliminating the risk of infections and allergies, and extending the use-by date. From here, I would like to introduce some of the developments we are currently working on. We have generated HLA no iPS platelets by gene editing and confirmed their circulation after administering of HLA no platelets using refractory mouse model in which anti-human HLA antibodies were administered. Normal HLA-expressing platelets are eliminated immediately after administration in presence of anti-HLA antibody, but HLA no platelets were found to circulate in the body even in the presence of antibody. In addition, it was found that HLA no platelets are not attracted or attacked by human NK cells due to the loss of HLA, which is a common concern. Thus, we have obtained evidence that the HLA no platelets that we are developing could be useful for transfusion refractoriness. One of the most important aspects in the commercialization of iPS cells is the reduction of production cost. We have found conditions that greatly accelerate the enlargement of MKCL size at maturity in order to increase the number of platelet released per MKCL, which is the most important factor in reducing production cost. Process 2 promotes MKCL polypolydization and cell enlargement. The number of platelets produced under this condition increased 8 times. Based on these results, we are now working to improve the manufacturing process on a practical scale. We believe that process 2 can reduce production costs to at least 1/5, and we are aiming to reduce it to less than 1/10 through further improvements. If we can achieve this, we believe that we will be able to deliver value-added iPS cells to patients anytime, anywhere, and to anyone. As a member of Sysmex Group, Megakaryon will continue to develop a new world of platelet transfusion. Thank you very much for your attention.
President AKAMATSU, thank you very much for your presentation. The next speaker is sitting next to him, who is President MASAYA Matsumoto of TWOCELLS. Thank you very much for giving me the opportunity to present today. My name is Matsumoto from TWOCELLS. TWOCELLS was founded in 2003, and it is our 22nd year. We are doing the development of regenerative medicine products using allogenic MSC, specifically culture in serum-free medium allogenic MSCs through supply system of donor tissue and stable supply of products with the stable quality with the platform of allogenic MSCs and long-term storage with our freezing technology. And as I'm going to show you later on, our strength include phase 3 clinical trials based on data for quality and performance. I'm originally from research, but last November, there was the founder and the chairman, and there was also the second generation president, but I now serve as the third president of the company. The technology I'm going to introduce today is the gel sheet allogenic cartilage regeneration product, GMSC1A, and the second one is for renal failure, serum culture, highly effective with a cell suspension, GMSC4. In order to have the mass production, serum-free media, know-how on manufacturing, and clinical development are our area of strength. With allogenic cell, we are calling as the guaranteed MSC, such as a GMSC, which will help to have the products that are mass producible. So for the MSC, compared with the ESL and iPS cell, they have less concern over canceration, and in order to commercialize that, we have less concern for safety. And there are some which have been already launched in Japan as regenerative medicine products. This is the first strength we have. This MSC, in order to culture in high quality and mass production, we have STK series for serum-free medium. This is to reduce the risk by using the own serum and traditional animals, and it is possible to retain high level of differentiation and proliferation. And this allogenic, this is the second strength of allogenic. MSC that we handle is through supply system of a donor system tissue, which is allogenic MSC. It is essential to have allogenic for the commercialization of regenerative medicine. If it is autologous, the inspection and manufacturing costs will be high, and we also have the concern for the collected quality of cell. So it is important to have allogenic as one of the solutions. And this shows the supply chain of GMSC, and the donor synovium is received from the patient at the time of surgery, and what is manufactured as a product will be used as necessary after freezing and storing. This enables to supply homogeneous product stably, such as a cell suspension and a gel sheet. These are possible to be supplied. A 3D structure gel sheet is possible to be stored for a long time, so stable supply is possible. In various areas, we have patents and know-how to protect this technology. This GMSC1A was presented by Professor Nakamura earlier, and through high-density culture, it is possible to structure 3D structure with cell and extracellular matrix. Just by fixing to cartilage injury site, it is possible to attach easily, so there is no need for suture and fixed will sealant. This is a very simple surgery. And now, I would like to show you the clinical study data that we have conducted. In comparison with microstructure, we have conducted a third phase clinical trial in Japan. In comparison with a microfracture, you see the pictures, and GMSC1 is attached to the injured site. And at this point in time, the transplantation is completed. Fixing is possible in a matter of a few minutes. So it is a simple transplantation. Microfracture and GMSC1, you see the MRI pictures as you saw earlier. The cartilage is clearly regenerated. Microfracture will have the holes in the subchondral bone of the cartilage, but in the GMSC1, you see clear regeneration of the tissue. This shows the scores of what I explained. In the past, to a certain extent, microstructure will repair the cartilage, but we are able to have significantly greater difference through the clinical study. Now, you see the cartilage regenerative products used both Japan and overseas. Many of them are still autograft. We are allogenic. It's a 3D structure, and simple surgery are our strength. So we would like to leverage the advantage to promote our development. So what I showed you is related to GMSCA1, and those are gel sheet. And I would like to also talk about a GMSC4 cell suspension for kidney. This is cell suspension product in development. There is fibrosis, which is the mechanism of action for CKD, and MSC will have the anti-inflammation and also anti-fibrosis effect that is known. So TGFB1 and alpha SMA are known to be expressed, but it is also known that it can be suppressed by MSC. As you can see, our cells are shown by the blue bars, and compared to the neighboring orange and gray, they are different cells. So compared to serum, TGF-beta and alpha SMA can be suppressed according to the animal study. So these cells which have been cultured by our MSC, we know that the TSG6 expression is enhanced. and we already have the patent. So by leveraging that for a kidney, we would also like to develop for GMSC4 as our products. Guaranteed GMSC is our mainstay product, and we would like to continue the development of this product. And for the patients who can be cured with our cells, we would like to deliver the product. Thank you very much for your attention.
Thank you very much, President and CEO, Dr. Matsumoto. Next, I would like to invite Dr. Keiichi Fukuda, founding scientist and CEO, Heartseed.
I am Keiichi Fukuda, a founder and CEO of Heartseed. We are developing the world's first remuscularization or cardiomyocyte replacement therapy for a severe heart failure patient. It is a video of the actual heart of the heart failure patient. It is very weak in movement and enlarged than normal. Please listen to me. The heart becomes less powerful to pump and getting enlarged. Please listen to me as if it were your heart. You have been very severe heart failure and have been on a drug medication over 10 years. But it is only getting progressively worse. And the heart becomes less powerful to pump and getting enlarged. You are facing the two choices, a heart transplant or mechanical heart. The existing therapies are no longer sufficient. You have to make a choice about your life. Please watch the video on the right. We want to bring it back to this kind of heart. Even it is not 100% healthy, the heart becomes smaller, the pumping function will increase, the useful movement will be restored. You wish there is such a therapy, since the existing therapy cannot achieve this. You know what? In fact, this is actually the case of the clinical trial, which Heartseed is progressing now. The video you are watching is the first video in the world that demonstrates the effectiveness of the regenerative heart muscle transplantation. Heartseed is a startup company that develops the world's first iPS-derived cardiomyocyte replacement therapy. I established Heartseed in 2015 when I was convinced that we can implement our technology to the society. I drive the business forward with a management team that are well versed with global biotech business. We have received global recognition. On the left, Heartseed was named one of the top 5 cardiovascular drug startups in the world by European Status Insight magazine. Since we initiated the first in-human clinical trial of the heart muscle transplantation, Nature magazine highlighted the success of the first case of HS001 clinical trial and published a special article. So, we are attracting great attention worldwide. Since the cardiomyocyte cannot regenerate by themselves, if myocardial infarction occurs, the number of cardiomyocytes will decrease, eventually causing heart failure. The heart becomes enlarged to maintain cardiac function, leading to the repeated hospitalization and eventual death. There are about 65 heart failure patients in the world. And it is said that the heart failure pandemic is coming in developed countries with aging society. I'd like to show the image of the world's first cardiomyocyte replacement therapy. We are developing the regenerative ventricular-specific cardiomyocyte from healthy donor iPS cells and use it to create cardiomyocyte clusters called cardiomyocyte spheroid. Cardiomyocyte spheroid is composed of approximately 1,000 cardiomyocytes. And by forming cardiomyocyte, the transplantation was 20 times more efficient to be engrafted than the discrete one. The transplanted cardiomyocyte grow in size after transplantation and can replace the missing heart muscle. This is our product pipeline. HS001 is iPS cell-derived cardiomyocyte spheroid derived from healthy donor iPS cells and is applied to the patient and in conjunction with open heart surgery. The final clinical trial LAPIS study is underway in Japan. HS005 uses the same cell as HS001 but different administration method by using less invasive catheter. In Japan, Heartseed is in charge and aims to submit clinical trial notification within 2025. In overseas, Novo Nordisk is in charge and they began consultation with authorities in the United States or European countries. HS040 is cardiomyocyte spheroid derived from patient-owned iPS cells and is targeted to the patient who cannot use immunosuppressant or wealthy individuals. This program has received government subsidies that will accelerate clinical development. We formed alliance with Novo Nordisk, the world's second largest pharmaceutical company by market cap. HS001 and 005 cardiomyocyte derived from healthy donor iPS cells are in the scope. The consideration is $598 million US. In Japan, two companies shared a profit by 50-50. In overseas, Novo bear the cost of the clinical trial, manufacturing, and sales. And Heartseed will receive royalty income based on overseas sales. Novo Nordisk has sales channels to 168 countries, and Novo have established know-how for the regulatory approval and sales strategies in each country. We can take advantage by this alliance to capture top global market share. Finally, I'd like to talk about myself. When I became a cardiologist several decades ago, I was faced with patients with many heart failure patients who have no treatment available. With great frustrations, I promised myself to save them through regenerative medicine. Since then, I have mastered science and come this far with a number of world-first research achievements. Based on the Heartseed mission, open the door to the treatment of heart disease through regenerative medicine, I'd like to realize the social implementation of the world-first heart failure treatment. Finally, Heartseed went public at the end of July, so I hope you will pay attention to us. Thank you very much for your attention.
Thank you very much, Dr. Fukuda. Next, from Orizuru Therapeutics, I would like to invite President Nonaka. First of all, to Nippon Keizai Shimbun and Kenta Fitzgerald Securities, Mr. Murata, and stakeholders, thank you very much for giving me this opportunity. I'm Orizuru. My name is Nonaka. I would like to talk about our business and the pipeline business. So Kyoto University and Takeda conducted a 10-year research program, and Kyoto University Professor Yamanaka led the way, and it was established as a bio company in 2021. Our goal is to have a bio company into a product, and at the Shonan Bio Park, we engage in business. In addition to the pipeline business, to have the platform and pipeline is our important mission. This slide shows our governance. First, up to Series B, we completed the funding, and in pipeline business from AMED, we received a subsidy. And since the establishment, we succeeded in procuring over 10 billion yen funding governance. So we put regenerative medicine into society. This is our social mission, and the foreign affiliated investors are also the investors as shown on the left. We have diverse shareholders, and we have independent directors in large numbers in our board, and we have a scientific advisory board where the Professor Yamanaka is a member. Now, briefly talk about my career. In 1990, I graduated from medical school, and as a surgeon, I worked in clinical practice for 10 years, of which 2 years I worked at the Texas Medical Center doing the research of the artificial heart. And after that, I worked for foreign affiliated company in R&D area, and I served as clinical leader and clinical expert as well as head of the R&D. As a clinician, at the ICU and CCU, I treated the severe heart failure, and at pharma company, I led as a clinical lead, and I was involved in development and launch of DPP-4 inhibitors. And I feel very close to those pipeline, and I participated in our company's business in order to sum up my life career. This shows the regenerative medicine challenges. There are 4 challenges: a clinical application and commercial application. So how can we secure high quality? And are we able to provide this treatment in an affordable way? So regenerative medicine hope that the transplanted cells should stay for the rest of the time. And the safety is also important for each challenges. As you can see on the right-hand side, we have a results approach. So I would like to explain by using the following slide. So our core technology is that we have the preparation technology. We have 2 products, cardiomyocyte and islet cells. And this shows the differentiation slide. So bioreactor is used, and with the differentiation technology, very mature, and cell implantable cells are manufactured here. A part of the process is completely closed. I will skip the detailed data, but for both products, CMC consultation with PMDA is already complete, and the manufacturing process and specification has already been confirmed. In this bioprocess, scaling up contribute to very much for the scale up. At maximum, the production cost can be reduced by 1/10 according to our calculation. Next, result of non-clinical study. So pancreatic islet program and myocyte program, we have OZTX-410-556. So major non-clinical studies have already been completed. So in the disease, animal model, and the carcinogenicity and other studies, all criteria have been met so that the clinical study can be conducted. So over 95% cell purity can be produced. That has resulted in the safety of the product. In parallel, under the JMPA process that was agreed on PMD with PMDA, we are doing study. This slide shows the long-term efficacy using islet. In the diabetes animal model, after the implantation, you see the blood glucose level normality, which continues for 24 hours. On the right-hand side, you see the pathological results. It shows the structure of islet. Not only beta cells, but also alpha cells are confirmed in the structure. And the pancreatic islet structure, and this is very similar, meaning that not only insulin but also glucagon secretion are also done. So physiological blood sugar control can be conducted, both alpha and beta. Next is a long-term efficacy. In the more than 10% of improvement of LVEF was observed, which continued for 26 weeks. On the right, you see the pathological sample in 6 months. In LV myocardium, you see the graft, and grafted cells is considered to be related to the LV function. This is a portfolio. As explained in the previous slide, after the 2 leader products, we are working on next generation as well. In order to be successful for the cell implantation, you need to improve the method, and with the autologous control is also a challenge that we need to prioritize. In order for us to succeed in this, experienced researchers and with companies with technology, we need to collaborate. There are not so many things that we can disclose, but high-quality collaboration will be the key for research and development, and the presence at the Shonan IPAC is the key for that. So this is the CEO commitment that we can conduct further development. Orizuru Therapeutics is a young and active company. All the employees are proud in the iPS technology, and we have a passion to realize them into society. We want to maximize collaboration in and outside the company, and each employee want to have a harmony to realize the iPS technology to put into the practice in society.
NONAKA 社長ありがとうございました。So we go on to the next speaker, who is Mr. Daniel KEMP from.
Thank you, Dr. Nanako.
Mr. KEMP, please.
Konnichiwa, and thank you to the organizers for inviting me to this great forum today. The creation of CAR T-cell therapies by Carl June about 10 years ago transformed the treatment paradigm for many blood cancers, effectively creating a cure for some of these previously unchallengeable indications. Much more recently, Georg Schett in Germany showed that CAR T-cell therapies could cure autoimmune diseases, and not just one, but maybe several autoimmune diseases, really opening up the potential for CAR T-cells and cell therapies in general to be a foundational part of the future of healthcare. And I think everybody here talking about regenerative medicine agrees with that premise. Those commercial products that were spoken to earlier today, the CAR T-cell therapies, are autologous, so they are derived from the patient's own blood. And that brings its own challenges in terms of the time it takes to create the product, the cost of the product, and the inability to translate this into a global therapy. So really, only in certain countries like Japan, US, and in Europe, can autologous cell therapies really be created and given to patients. So the future is allogeneic cell therapies, off-the-shelf, donor-derived cell therapies. And at Shinobi, we have focused on 2 of the biggest issues with allogeneic cell therapies. Number 1 is manufacturing. Manufacturing of cell therapies is one of the biggest challenges in the space. I think we'll probably talk about this in the panel afterwards. But essentially, to create a product with a cost of goods that's sufficiently low and also consistent with the challenges of donor-derived cell therapies, being that each donor is different, we believe that an iPSC platform solves many of those problems. The second issue with allogeneic cell therapies is persistence. So when one is giving a cell therapy from one donor to a patient, the patient's immune system immediately rejects those cells. And so one of the biggest challenges with cell therapies is to protect the cell therapy from the patient's immune system. And there are now emerging genetic approaches to cloak the cell therapies. And this is, again, another focus of Shinobi Therapeutics, at how we believe that we can really compete in this space. The two technologies that address these two challenges came from our founders. Shin Kaneko, based out of Kyoto University at Saira, worked with Shinya Yamanaka for over a decade now, specifically focused on iPSC-derived T cells and NK cells to treat patients with immune diseases and cancer. On the other side, in terms of the hypoimmune technology to cloak the cells from immune rejection, Toby Doise at UCSF, who's a transplant surgeon, has been working for about 15 years to develop ways to make hypoimmune cells. He's also a founder of a very successful company in the US called Sana Biosciences, and we're now working with his next generation of hypoimmune technology. You can also see here with Shin and Toby how our name actually came to be. So Shin and Toby together equals Shinobi. And it was really just a really nice coincidence that obviously Shinobi is Japanese for ninja, which we also like because it really speaks to the stealthiness of our cells in the immune system of the patient. Both of these scientific founders are intimately involved in our company, helping us advise and move things forward, and also to help manage our company from both the US side and the Japanese side. They both publish very much, and so most of the technology that we're developing is already in the public domain in terms of publication as well as IP. But it goes to show that we're a very straightforward company that is working on technology that we believe very strongly is validated. Our pipeline is very focused, but also aspirational. We have programs that are targeting solid tumor indications as well as autoimmune indications. And we'll be developing iPSC-derived alpha-beta T cells as well as iPSC-derived NK cells. We, in fact, do have a program in the clinic that we really see as a proof of concept technology. And every program we do take into the clinic will be increasing our validation of aspects of our technology platform so that we can move forward and build out what we believe is going to be the foundational platform called Katana, where we can plug and play different CARs or CAR TCRs to actually expand our indication spectrum. We are expecting to get into the clinic in 2027 with at least 2 of our products, targeting IND or PMDA at the end of 2026. One of the biggest strengths of this company is the fact that the technology and the team is global. Out of Kyoto University, we strongly lean on a team of about 33 full-time staff, research scientists, working very closely with the academic team of Shin Kaneko's lab at Saira. And we intend to put all of our products first in human in Kyoto and or Tokyo and leverage the facilities at Kyoto University, including a GMP manufacturing facility, as well as a first-in-human clinical trial site. We believe this is a very cost-effective and efficient way of testing our products in the clinic as soon as possible. We do also have a team in the US based in South San Francisco, for further development and expansion of our programs into phase 2 and beyond, we absolutely require to go clinically beyond Japan. And so the US is clearly an area where we need to have a footprint and we need to have a focused expansion, particularly around clinical and regulatory. Having a global company like this also provides us with the opportunity to bring in investors, not only from Japan, but also from the US, from Europe, and the world at large. And we've seen this in the way we built our syndicate from our first Series A round. Currently, we're at about 50 employees. We're looking to grow that, but not significantly. As I'm sure you're all aware, it's complicated and it's long-term to develop cell therapies. We're still at the very early stages of where cell therapies need to be off the shelf and globally utilized. Our team, as you can see here, is also global. The leadership team, who work both in Japan and the US, are pretty equally distributed. I do want to point out that our scientific advisory board, I think, is extremely talented and world-class, with Carl June, the inventor of CAR T-cell therapies. Georg Schett, as I mentioned, discovered that CAR T-cell therapies are also effective in autoimmune diseases, and Katie Rezvani, who developed the first CD19 CAR NK cell product that was shown to be successful. So we have a team that is very strong and continuing to get stronger as we move the company forward, both in its presence in Japan and the US. And finally, I want to also speak to the point that this can't be done as a company that is just insulated in and of itself. We have many different partners that help us progress our company and will continue to grow and partner with great partners like Panasonic, who are helping us think about closed system manufacturing, an area we think is going to be very, very important in the future, as do the Panasonic scientists. A team in Sweden, a company called NONAKA, who specialize in TCR manufacturing and design. We're working with those guys also. Very, very importantly, we work closely with AMED, and we're awarded a very large grant at the end of last year, beginning of this year. We take this very soberly and intend to succeed in the program and the platform for which AMED has very generously supported the company. And at the bottom, you can see we raised a significant round, a Series A, and the point here I want to make is that these investors are truly a global syndicate. We have investors from the US, from Europe, and several from Japan as well. And so this is the way we intend to build our syndicate further. It's very important because the cost of developing cell therapies is quite intense, and it takes more time. We will need repeated series of investment at a good level in order to really succeed as a company. And again, this is one of the biggest challenges of every company here at the table, is the continued buy-in from a very strong and supportive syndicate of investors. So I'm going to stop there and thank you for your time. ありがとうございます。
ありがとうございました。
Thank you very much. I think all the speakers have spoken longer than the time allocated for each, so we do not have a lot of time for Q&A, but I'd like to ask you, each one of you, just one question each. First, Dr. AKAMATSU of Megakaryon, please. Regarding technology to produce platelets, and you have been working on this technology for many years, and you had some technical difficulties and also funding difficulties as well. Now you are a member of Sysmex group. So by joining a group of companies, do you think that that has made your life easier in terms of funding? You do not have imminent difficulties? Or do you think that you have lost some kind level of flexibility? So the environment you are in can be called as an ideal environment you have aimed for. So could you please respond to this question briefly? Right. like in the case of other startups, we had difficulties in funding. Well, in the first phase, we were blessed with lots of funds, but as the exit, IPO was something that we were aiming at. But as you said, platelets, compared to other cell therapies, 10 to the power of 11. So over 100 billion needs to be produced. Therefore, to produce in that magnitude was quite challenging, more than expected. And we have joined in the Sysmex group now. And one thing, as you know, Sysmex, in the hematological area, they are expert globally in the reagent in hematological area. So there is affinity with them. So technically speaking, we are going to continue collaboration with the group companies. But for now, inclusive of various physicians, we needed to strengthen and utilize the network. So in that sense, and I think that the combination of us was quite good. And funding, as we have become a subsidiary of Sysmex, it's not that we don't have to care about the funding, but we have this opportunity to be called to speak, to find the venture capitalists as well as investors going forward. We like to explore and finding those in the future. Thank you very much. And TWOCELLS, Dr. Matsumoto, your technology has a broad range of application. So by launching your products in order to increase your revenue and earnings, I think that it will take some more time. What about your strategy? Right. Currently, we do not think that we have ample funds at hand. Of course, IPO can be one option, and partners such as pharma companies and also funding through equity issuance and so forth. Including all these, I believe that there are a lot of various options. So first, we have to put most focus on technological development in order to develop the technology or products that can be sold. But inclusive of such forum like today's, domestic and overseas investors, and how we should consider the approaches to get funds for development, we are exploring a wide range of opportunities as such. Thank you very much. Do you think it is going well, smoothly?
Yes, the fact that I'm invited in this kind of venue represent that. So in Japan, also, including the government policies, the ecosystem was explained about the funding, and we receive a lot of support. As for development status, we are at the very late stage. So in the late stage, as was mentioned, in the early stage, it's easy to receive funding, but at the late stage, the amount that we have to procure and the level of easiness to receive funding becomes more challenging. So it will be rather challenging to receive funding only in Japan, but we have to think about the overseas as a funding source moving forward. Thank you. Next to Heartseed, President Fukuda. So late stage is the phase that Heartseed is in, and you started a clinical study. As for heart, there are other companies that want to treat heart with regenerative medicine. In order for you to be a very prominent company in this area, how creative have you been? And Novo Nordisk, and you align the fact that you have a big company behind you is a strength for you. And how did you realize that? And to make it a sustainable relationship, what is required, and what are your thoughts on this? At the initial stage of clinical development, so open chest and with especially made needle implantation to the heart was done. But in order to go to the volume zone, you need to conduct clinical trial using a catheter. So at the early stage, we wanted to start clinical trial using catheter, and we are making preparation for that. As for alliance with Novo Nordisk, a few years ago, at the U.S. conference, with some big global farmers, we had a meeting, and finally we decided to work with a novel. Concluding contract with large pharma companies is a big challenge for a Japanese venture, but a certain milestone amount will be provided on a regular basis is a benefit for us. And when we are at the late stage of venture in Japan, it is rather difficult to procure a fund for late-stage venture in Japan, and that is widely known. But by having alliance with a big pharma, we are able to receive milestone payment on a regular basis, and we want to have a breakthrough of this kind of challenges. Thank you very much.
野中社長、お話の中で
So, President NONAKA of Orizuru Therapeutics, you spoke about Shonan Park, and we are working on that. And I believe Shonan I Park is also one of the possibilities. And can you talk about the advantages and what do you think is lacking with this Shonan I Park? Thank you for the question. To be candid with you, the motivation of the researchers is very high. The facilities are very new, and it's nice to work as an environment. And compared with the global ecosystems, Shonan I Park facilities are world-class. As to what we are going to do in the future is going to be basically on the track record. Unless we have the companies who will have the social implementation, it will not be viable. So we would like to make efforts to do so. Thank you very much. Then Shinobi Therapeutics CEO, Mr. Daniel KEMP, as I listened to you earlier, there's nothing that you need to be afraid of. You don't have issues. Basically, your challenges are about to be solved. And beyond that, what do you think is necessary? Can you cite one or two points?
I would not say that everything is solved. I think we're not going to be in the clinic for 2 years. And, our cash runway doesn't take us all the way to the clinic at this point. So we need to find creative ways to continue to raise money, to strengthen the team, to move the programs forward. And we have good strategies to do that. But things are very different in 2024 than they were in 2021. So my last job as a CEO for a company called Wugen in the US, we raised $170 million in 2021. I think the technology at Shinobi is significantly better, but it's so much more difficult now to raise money. And it's also very, very important and somewhat expected by venture firms to be getting into the clinic as soon as possible. Venture firms do not want to put money into a company with a cash runway that doesn't reach the clinic. And I completely understand that. I think we all do. But with cell therapies, there is a timeline that forbids you from going straight to the clinic like you can with small molecules and even with biologics. So, I think we still do have challenges just like everybody else on the panel here in terms of continuing to replenish our funding, keep our investors excited. And of course, then there are also external events that are going to happen. There are other cell therapy companies who are reading out clinical readouts, and really great readouts could potentially help us, and poor readouts could potentially hurt us. And so these are very unpredictable. These are the things that worry me a little bit more, is what's happening in other companies that actually do have read-through as to what we're doing at Shinobi. But my final point is really that I do believe that we have the most exciting and robust science. And I do also believe that iPSCs will be fundamentally stable of cell therapies in the future.
Thank you very much. Although I wanted to continue asking you questions, but I know that the time is pressing. If there is any question, just one question can be taken from the floor. Are there any? If not, well, you may have opportunities to ask questions individually later. Thank you for your participation. All, please give them a round of a big applause. Thank you very much, everyone. Now, please return to your seats. Dr. Akamatsu, Dr. Matsumoto, Dr. Fukuda, Dr. Nonaka, and Dr. Kemp, Daniel Kemp. Thank you very much.